Right, good evening, everybody. As principal, it's my very happy duty to introduce this inaugural lecture, the No.1 Ladies' Disease Detective Agency, from mortuaries to marine mammals, to be delivered by Professor Ailsa Hall of the School of Biology. This compelling title, drawing upon the Scottish crime series by Alexander McCall Smith, is just as unconventional as Ailsa's career itself has been. Quite how Ailsa's academic life has paralleled the Botswana-based Ladies Detective Agency, I am unsure, but I'm much looking forward to hearing about it when Ailsa delivers her lecture in a few minutes' time. I suspect Ailsa's career will feature much less red bush tea and a great deal more formaldehyde. So, as the title of tonight's lecture suggests, Ailsa's experience has been eclectic and wide-ranging, but it is this which has enabled Ailsa to develop unique insights and dynamic thinking and has resulted in the professorial appointment that we mark today. Ailsa undertook her first degree study at the University of Bradford, graduating in 1984 with a Bachelor of Science in Environmental Science. After a year spent working in the sewage industry after graduating, Ailsa joined the University of Birmingham's Institute of Occupational Health as a research assistant and PhD student, and she graduated there in 1989 with a thesis entitled Mortality and Morbidity of Pathologists and Post-Mortem Workers. This thesis focused upon the pathologists and morticians who interact with bodies of the deceased and who, in so doing, come into contact with infectious agents. It was of such significance that it informed and inspired a code of practice, a new code of practice, for the prevention of infections in post-mortem rooms. Towards the end of her PhD, Ailsa was appointed a research fellow at the Institute, where she organised the Occupational Health Master's course and published a paper entitled The Epping Jaundice Outbreak, a 24-year follow-up. So even then, she was producing evocative paper and publication titles. The standard of excellence, the capacity to produce impactful research, the knowledge of academic and industrial environments, and the ability to work across disciplines that Ailsa developed during her 9 years as a student made her a perfect fit for the Sea Mammal Research Unit, or SMRU, which was then based in Cambridge and which she joined in 1989. It was there that Ailsa began studying marine mammals. In 1988, hundreds of harbour seals were killed by infectious disease, so Ailsa was hired to investigate the cause of death. While undertaking this work, Ailsa was intrigued by the way that chemicals sequestered into the fat stores of seals were affecting their immune systems and leaving them open to disease. Ailsa's research ever since has focused on the impact these compounds have upon marine mammals, how they affect immunity and reproduction, and the consequence of these pollutants on animals in the natural world. It is ever more important and urgent work in the light of the climate crisis which we all face, and Ailsa's work has pioneered protection of the natural world. SMRU relocated from the Cambridge Antarctic Survey to the University of St Andrews Scottish Oceans Institute in 1996, and Ailsa has been part of our university for over 2 decades since. During her time with SMRU and at the SOI, Ailsa has held various roles. She joined as a research fellow and was later promoted to reader. In 2008, she became deputy director, and she was appointed its director in 2012. As director, Ailsa provides support and infrastructure to 8 academic staff members and has overall responsibility for the SMRU Instrumentation Group, comprising 9 engineers and with an annual turnover of around £1 million. SMRU owes much to Ailsa's expertise and strategic vision and the work she undertakes to ensure that it continues to be a foremost place for the study of marine mammals anywhere in the world. SMRU's prestige is part of why the university invested £16.5 million into the development of the new SOI building, which opened so wonderfully in September 2019. These positions of responsibility have been held in parallel with an accomplished research and publishing career. Ailsa has around 150 publications in total, including 5 book chapters and many more contract research reports. Ailsa is currently the principal investigator on several key projects. These include NERC's National Capability Marine Mammal Research Programme, a 10-year rolling programme, as well as the Scottish Government-funded Marine Mammal Scientific Support Programme, a 5-year programme. Ailsa has used her research to contribute evidence-based science to policymakers on national and international stages, and to the numerous groups of which she is a member, including the Scottish Association of Marine Science and the International Whaling Commission's Scientific Committee. Ailsa's international distinction has been recognised by visiting scholarships at the University of Tokyo and at the US National Marine Fisheries Service. In addition to her distinction as a researcher, Professor Hall is also an inspiring colleague, a widely respected leader, and a much valued teacher to her students. And in her role as director of SMRU, Ailsa demonstrates exceptional managerial, financial, and organisational acumen on a daily basis. We feel fortunate to have her as part of our community, and it's great to celebrate her accomplishments this evening. After tonight's lecture, a vote of thanks will be given by the Acting Dean of Science, Dr. Jonathan Isberner. You're then all warmly invited to a drinks reception in Lower College Hall. So please now join me in welcoming Professor Ailsa Hall to deliver her inaugural lecture, The No.1 Ladies Disease Detective Agency, From Mortuaries to Marine Mammals. Ailsa.
Thank you very much. Thank you, Principal. So, who are these disease detectives? Well, we might generally think of physicians and, in the case of animals, veterinarians as being the main diagnosticians and investigators of disease occurrence. But there are other disciplines involved in disease detection, and that includes the epidemiologists, who are interested in the study of disease on populations rather than individuals. So this might be tracking and predicting the spread of infection, such as in the current coronavirus outbreak, but also epidemiologists are interested in the causes of disease, which even in the most obvious cases of infectious disease are indeed multifactorial. Well, there's another group of discipline involved in disease detection, and that's the pathologists. And that's where my career in epidemiology began, with a study of health and disease risks for this population. As Sally said, I got my first degree and then a couple of smelly jobs in sewage and waste disposal, and then got the position at the Institute for Occupational Health on a Department of Health funded study to look at the disease and occupational disease hazards for this group of workers. At that time, the code of practice for the prevention of infection needed updating, and it was at the time when the HIV virus had just been discovered, and so it was the worry that this was an occupational risk for these health workers. Many of the mortuaries and autopsy suites, particularly around the West Midlands, look like this, with these ceramic post-mortem tables, really hard to clean surfaces and poor ventilation, and I visited quite a number of these, so I can verify that. We, in this study, investigated bacterial contamination and hygiene methods during many autopsies carried out throughout these facilities in the West Midlands, and also a morbidity and a sickness absence study in mortuary technicians, but also a mortality study of British pathologists. And our cohort, our study population, were members of the Royal College of Pathologists, which in those days was housed in Carlton House Terrace in central London. And I spent many hours in the library there, extracting details of over 4,500 members of the college from 1974 to 1987. And in those days, there were no computerised records, so this was quite a laborious task of looking at membership lists and ledgers, requesting the death certificates for those those pathologists that had died. And what we found was that there was a significant increased risk of suicide amongst this population. We observed 13 and expected 5, but also an increased risk of brain cancer deaths. We observed 6 and expected 3. And when we looked at these cases in more detail, we found that many had disseminated tuberculosis. Tuberculosis is a known notifiable disease for pathologists, and the theory was that the tubercules in the disseminated had migrated into the brain and caused a focal for tumorigenesis. Well, I'm pleased to say that this body of work did result in an upgrade of the code of practice for prevention of infection, and also many modern autopsy suites look like this now, with stainless steel downdraft autopsy tables, easy to clean surfaces, and better ventilation. So I submitted this body of work for my PhD, and then was landed the very interesting and very novel investigation and case of disease detection known as the Epping Jaundice. And this began in February 1965 when a medical student working at St Margaret's Hospital in Epping fell ill. He lived in Harrow, which is outside London, quite away from Epping, and he had these severe abdominal pains and a jaundice, a yellowing of the skin. He, still feeling pretty poorly, went back to work on the Monday and saw this local GP, Dr. Harry Kopelman, who was quite alarmed by his symptoms, admitted him to hospital. Harry then went to do an outpatients clinic at a local hospital in Harlow and found that a local GP's wife there had similar symptoms of jaundice. He then telephoned around a lot of other GPs and found 6 simultaneous cases just in the Epping and Ongar area in the period of a few days. The clinical presentation was quite unusual with this jaundice, and at first the investigators thought they were dealing with an outbreak of hepatitis. The hepatitis virus had just been isolated, and they were quite alarmed that this was going to be a major viral outbreak. But the clinical presentation did not fit the hepatitis presentation. In total, they had 84 cases over a period of a couple of months, and one case in particular led to a breakthrough in the investigation, and that was that the medical student's wife also went down with the jaundice some days after he did, and the only link between her and the Epping area, so they could find no links, as I say, with questioning amongst the cases until this one, was a loaf of wholemeal bread. Now, bread is not usually known to be the cause of food poisoning, so it's no wonder they didn't really pick this up at the time, but they went back and questioned all the cases, and indeed they had all bought the bread from the same bakery or the branch in Epping and Ongar. They then realised they were probably dealing with a toxic event. One of the ladies, one of the cases, had eaten some of the bread, found it to be very bitter, left it out for the birds. The birds hadn't eaten it, so they managed to go and collect some of this bread, which was pretty soggy by then, and feed it to the birds, feed it to the mice, and the mice went down with jaundice. So they knew they had the link between the cases and the exposure. They went to the bakery, they could find no contamination evidence there at all. So they then focused on the transport of the flour from the mill to the bakery, and they found that it had been transported in a van with containers from a large chemical company. And on questioning of the van driver, eventually admitted that one of the chemical containers had indeed fallen over and spilt, contaminated the flour. He hadn't noticed or hadn't cared, sent it to the bakery, and it was made into the bread. The flour was contaminated with 2-part epoxy resin hardener, we're all familiar with, in the glue, which contains this chemical methylene dianiline, or MDA. And since this time, it has been classified as a proven animal carcinogen. And I have to say, at the time, all the cases recovered, but it was our job then, 24 years later, to follow these cases and find out if there was any long-term health issues. The 2-part epoxy resin is widely used in industrial settings, and particularly in those days in carpet laying, where they used it a lot for gluing down the carpet tiles. So we went to see Dr. Harry Koperman, got his original case notes, which was great, and talked about the background to the outbreak. We traced 68 of the 84 cases, 50 of whom were still alive, so I managed to interview quite a lot with health questionnaires. One interesting chap was the managing director of the Ford Motor Company, who admitted that he felt really poorly after a meal and decided that it couldn't possibly be the bread, so he ate some more bread and made himself even more sick. 4 of the cases had further episodes of jaundice, and 18 people had died, but the causes were unremarkable, except for one carcinoma of the biliary tract, which is quite a rare cancer. So in a group of 84, that was a significant finding. But since then, it's not been shown to be related to MDA exposure. So it was a celebrated case of medical detection. It was the subject of a radio play starring Bernard Leptin in the late 1960s. And it was taken up in the House of Commons, and legislation was introduced to prevent foodstuffs and chemicals being transported in the same vehicles. So, how did I get from there to marine mammals? Well, another mystery infection was occurring in the seals in the North Sea in 1988, and the Sea Mammal Research Unit advertised for an epidemiologist, and I was lucky enough to be appointed. And it was causing this severe respiratory infection in the seals, in the harbour seals, and a high case mortality, so lots of carcasses washing ashore around the coast of Northern Europe and the UK. A lot of alarmist headlines in the newspapers about what this was caused by, until it was discovered it was a virus, a phocine distemper virus, which is in the same family as measles, human measles. It's a morbillivirus. So we had the outbreak in 1988 and another outbreak in 2002. So we have 2 species of seal in British waters and in Europe. The harbour seal, as I say, the common seal, was the victim of this outbreak, but also the grey seal. Now, the grey seal then appeared to be highly resistant to this virus. It was exposed, it did have antibodies, and we know it was carrying some virus, so it's potentially an asymptomatic carrier, but it was not succumbing to the infection the same way as the harbour seal. So also what was interesting there was regional variation in the mortality amongst the harbour seal. And in those days, we didn't have such good estimates of the harbour seal population abundance and distribution as we do now. Here's a recent map of the abundance of harbour seals shown with the red dots. So we've got a very good understanding now of where our harbour seal populations are. But in 1988 to 1989, we didn't have such good data, except for maybe 3 populations where we had reasonable estimates. So in the Wash, the estimate was about 50% mortality, in the Moray Firth 15%, and in Strangford Lough 30%. So what was causing this variation in mortality despite similar exposure and antibody uptake? Well, as we've heard, one of the potential factors was that the animals are also exposed to these persistent organic pollutants. These are widespread industrial chemicals that were synthesised in the 1930s and used right up until the mid-1980s when they were banned, and their production and use has since been banned. But they're highly persistent, and everybody, I'm sure, is well aware of the DDT and chlorinated pesticides story with the effects on birds of prey, and the Rachel Carson Silent Spring book. But also the polychlorinated biphenyls, the PCBs that we widely used in capacitors and transformers and comprise a very wide range of different chemical compounds. And those were the chemicals that we were particularly concerned with. Now, these compounds are highly lipophilic, so they have a high affinity for fat. They biomagnify up the food chain, so they start at very low levels, perhaps in the water and the phytoplankton, but by the time they increase and get up to the top predators, they can be at sometimes very high concentrations. They're stored, therefore, in the blubber, in the subcutaneous fat layers of the marine mammals, and they're transferred to the offspring during lactation. So as the females mobilise blubber stores into a fat-rich milk, the chemicals are transferred with that lipid. So breeding females tend to have lower concentrations than juveniles and males. But we know these compounds are endocrine disruptors, so that means they mimic hormones and they can adhere to and bind to hormone receptors. So they have health effects in that way, and they particularly affect reproduction, but also immunity, and that's the link back to the hypothesis with the phocine distemper virus. If you have a compromised immune system, you're not going to be able to fight off the infection, you're going to be so resistant, and so that was the question that we were asking. And what we found was that the PCB concentrations in the harbour seals was indeed higher in the animals that died of PDV, the victims, compared to the survivors. The team went out and captured some surviving animals in the years after the outbreak and collected small biopsy samples to compare with the concentrations in the deads. And you can see that the female concentrations were lower than those in the males. But when we looked at this on a regional basis to compare with those mortality estimates, we did indeed find that difference still between the dead and the live animals, but there wasn't any obvious correlation and significant relationship with the mortality estimates. So we had some evidence that PCBs may have played a role in the PDV, but it wasn't particularly convincing. Some of the reason for that might be because we're not able to to control for all the confounding factors in this, particularly the amount of fat the animals have, their blubber stores. If they're low, the contaminants concentrated in those thinner blubber layers, and when the animals are fatter, the contaminants can dilute. So you get that difference because of condition. So one of the great things about moving up from Cambridge to St Andrews was the fact that we were then able to collaborate and interact with the academic groups here in St Andrews. And one such group was the Comparative Immunology Group at the then Gatty Marine Laboratory, headed up by the late Dr. Val Smith. And she and I worked together on some aspects of the immune function of seals. And one question we were asking ourselves was, well, if the grey seals are resistant to the PDV virus, are they also more resistant to the effects of PCBs? And so in an in vitro study in the laboratory, we exposed harbour seal and grey seal white blood cells, the immune cells, to different concentrations of contaminants of the PCBs to see how robust they were. And in this assay, respiratory burst index, this is an assay that looks at the ability of the cells to neutralise the pathogens. In the harbour seals, that was reduced, it was a lower capacity, but in the grey seals, there was no difference between the control group and the exposed group. And also in a phagocytosis assay, which looks at how well the cells can engulf foreign particles, again, the harbour seals, even at the low concentrations, are more susceptible to that impact on their immune system, and the grey seals are pretty bomb-proof. So what's the current position? Well, the PCBs are still circulating, they're highly persistent in the environment, they're not metabolised by the animals, and there's this legacy from mother to offspring, so they're going to be around for some time to come. Still in the wash we have relatively high concentrations, but also on the southwest coast of Scotland, and there is a PCB hotspot in the Clyde estuary, which is emanating out into the local environment, and that's perhaps why we have this high level, particularly in the males, and above the estimated threshold for these adverse health effects at about 20 parts per million. So if, and probably when, we get another PDV outbreak, this virus is cyclical, and the population now is susceptible to another outbreak. A lot of the animals that were alive in 2002 that survived and therefore had antibodies and protected themselves against another outbreak have died and been removed from the population. So this is now a susceptible population, and of course there are some individuals in there that could be even more susceptible to infection. But what about the impact on cetaceans? That's the whales, dolphins and porpoises. Well, if your immune system is compromised, then you're likely to be susceptible to any infection. So in conjunction with our stranding scheme, cetacean stranding scheme colleagues, we carried out a case control study to look at the infection risk and PCB exposures in the harbour porpoise. This is a classic epidemiological study design, a case control study. And our cases were stranded porpoises that had washed ashore that had died of an infectious disease, so bacterial, viral, fungal diseases. And our controls were stranded porpoises that died due to acute physical trauma, so boat strikes or perhaps bycatch. And that gives us an estimate of the exposure in the population that gave rise to the cases. And you can see the exposure distribution here on the right. Now, the effect measure that you get from these studies is the odds ratio, so that's a measure of average risk. So if the risk is the same in the two groups, then the odds ratio is 1. And an odds ratio of greater than 2, which is considered relatively high, occurred when the exposures exceeded that estimated threshold for health effects. So we had some evidence that indeed there is a disease risk for these animals, but most of the population here have relatively low concentrations below that 20 parts per million threshold, but certainly some individuals certainly do have the higher concentrations. And of course the other point here is we don't know what the true risk, what the true incidence of infection is in the population. So if there's a doubling of risk but the occurrence is quite low, then that risk is maybe not significant. But measuring true incidence of infection in a wild population such as this is almost impossible. But what are the population consequences? Well, we know we've got effects on pregnancy and offspring survival and immunity. So we constructed an individual-based model to see what impact this might have on a prediction. And to do this, we had to integrate the measured tissue concentrations, so the blubber concentrations, with a dose-response relationship to understand and estimate what the population consequences might be. And because we don't have data from the study populations, from the cetaceans themselves, as in with other human risk assessments, one uses a model species, and in this case we had data from mink. These are carnivores that also eat fish, so relatively good models. And we had some dose-response data from a number of studies in the 60s, 70s and 80s to understand how that shape of that dose-response curve changes with the increase in maternal tissue concentrations. You can see that the proportion of kits that survive decreases considerably as the contaminants increase. So we use this flow diagram here to show you how the model works. We have an annual accumulation of PCBs from the prey, and then this is a female-based model, so we're not bothered about the males. They breed with a certain probability, and they offload a proportion of their burden to the calf, and then we apply the dose-response relationship, which decreases their probability of survival depending on their mother's concentrations. And if that means the calf dies, she's removed from the population. If the calf lives, then she goes back round and is counted in that population. So we can add up the number of animals that are left at the end of each simulation. But of course we know that the immune system is also affected. So we added a degree of complexity here to look at the effects on immunity. And again, some dose-response data that we had for model species and looking at disease resistance in relation to PCBs gave us an estimate of what this would be affected. Now, if your immune system is compromised but you're not exposed to a pathogen, then maybe that's not a problem. So you have to be exposed, and then again, you have some probability of dying or living and going back into the population. So when we ran this model for some example species, here the bottlenose dolphin, using that surrogate reproductive effects dose response and looking at 50% of the population exposed to a pathogen, then there's a lot of wiggle in here. There's a lot of stochasticity, there's a lot of uncertainty in this estimate. But on average, that population will decline because the potential population growth rate that we estimated is less than 1. So over 100 years of simulations, that population will decline. But as you can see that some of the population trajectories in the simulations do in fact increase. So there is that degree of uncertainty in there about what exactly will happen. Well, in conjunction with our Danish colleagues, we also applied this to populations of killer whales. Now, killer whales are even higher up the food chain. They themselves feed on the seals and whales, and so they have some of the very highest concentrations of PCBs in their blubber. And we applied the model on different populations across the globe, from the low exposed populations here in Antarctica and Norway up to the higher exposed populations, including some individuals that we have seen very high concentrations here in the UK. And applying this model, a population growth rate of 1 is a stable population, so the low exposed animals populations are increasing or stable, and then as you get higher, obviously those populations will decline over time. Now this model has a lot of assumptions, a lot of caveats, and it caused quite a bit of discussion in the literature about the validity and the assumptions that we made. But that's fine, that's as it should be in scientific endeavour. But it also was picked up in over 80 news items and 2 policy documents. And to quote a famous statistician who said, "All models are wrong, but some models are useful." So if this has generated interest in these legacy contaminants and still identifying them as a problem, then that's no bad thing. Well, an organism's fitness is highly reliant on its ability to fight off infection and its immune system. And this is an energetically costly process. Your immune system requires energy. And so there's often a trade-off between energy required for a robust immune system and for other life history traits such as growth and reproduction. And there's a newly developing discipline called ecological immunology, which is looking at individual immune fitness and how that is shaped by an organism's not only its evolutionary history, but also its ecology and its current environment. And we were able to contribute a little bit to this new discipline in a study looking at immune activity and dynamics in an endangered but highly well-studied population of Galapagos sea lions. Now, we needed a population that had 2 colonies of the same species, one that was heavily impacted by humans and one on an uninhabited island. And here in Cristobal in the Galapagos, there's a lot of interaction between sea lions and humans and domestic animals. And on the uninhabited island, you can only access that with a research permit, so we're the only people to have been there for some years. And we found that the immune activity was indeed higher on the impacted colony. This is outside of disease, overt disease, it's the higher stimulation. So there is greater immunostimulatory pressure on this population. So the theory is that may have implications for the colony stability and for the individual immune fitness and emerging diseases in that population. So I think this is a good example of how our research, although it's based in conservation and management issues, can also contribute to ecological principles and fundamental understandings of processes in mammals. Well, the animals are not only exposed to infectious agents and contaminants, they also have to endure other insults, including these natural toxins. And I'm sure we've all had a dodgy plate of mussels now and again, and maybe a touch of diuretic shellfish poisoning, which is caused by one of these toxins. And they're produced by harmful algal blooms, which are on the increase globally. So increased nutrient runoff from the land to the marine environment, and perhaps climate change issues as well. And this was amply demonstrated in its importance in marine mammals with the mass stranding of California sea lions in 1998. Some hundreds of animals came ashore, and this was due to a potent neurotoxin called domoic acid and produced by a diatom of Pseudonychia australis. Now, in acute exposure in the height of the blooms, these animals were taking a lot of toxin and it was causing them to seizure and die. But there were also some chronic cases, so animals that appeared not to have acute effects or had acute effects and then recovered, but they were then behaving very unusually, out of habitat, shall we say, so in fields, and then causing havoc when the police were called and the animals would haul out on the police cars. This apparently wasn't an unusual phenomenon. So couldn't understand quite what the reason for that was until they discovered in the brain of these animals they have what's called a hippocampal atrophy. So it's a unilateral atrophy. This is a normal hippocampus of a sea lion brain, and this is an atrophied, a shriveled hippocampus. And this organ is really important for spatial awareness and memory. Apparently, London taxi drivers have very well developed hippocampuses. And so that was accounting for this unusual behaviours. Well, we are concerned about some of these harmful algae here in Scotland. We do have Pseudonychia that does produce domoic acid, particularly here in Orkney. This is in Scapa, monthly blooms of Pseudonychia from 1996 to 2016. And you can see in the summer months this data from Marine Scotland, where they have these large blooms on some years, which would then go on to produce domoic acid. And here's an image of a phytoplankton bloom on the surface of the water in Orkney. And in conjunction with this, we are experiencing a major decline in the number of our harbour seals. This is outside of a PDV outbreak, and over the last probably 15 to 20 years, we've lost, in some regions, maybe 70 to 80% of those animals. Not everywhere, say the West Coast, populations are stable or increasing, but on the East Coast, here in the Eden Estuary particularly, and in the Northern Isles, these populations have been declining. And the Sea Mammal Research Unit has been investigating a number of different factors which could be causing this phenomena, but of course I'm particularly interested in the toxins. And so we went and collected some fecal samples from the seal haul-out sites, the harbour seal haul-out sites. This is the excretory route for the toxins, and so you can use this as an index of exposure. And you can see for the populations, and certainly some of the populations that are declining, we have some higher concentrations. But this is occurring across Scotland, and we're seeing exposure in areas where populations are stable. And so we're currently carrying out a risk assessment to look at the toxins in the prey, to see if indeed this is a factor in this decline. This is likely to be a low-level chronic exposure. We're not seeing seizuring seals as in the sea lion case, but we know that may have immunomodulatory effects as well. Well, unfortunately, these animals don't just get infectious disease, they also develop cancers. It's quite rare for wildlife to develop cancer, but the carcinomas in the California sea lions are quite notable. It's a urogenital cancer, which is highly aggressive, and it causes the death of about 20% of the animals that strand and come into this rescue and rehabilitation centre, the Marine Mammal Centre in California, in San Francisco. And you can see in this image the metastasized tumours. There's been many causal factors investigated since the 1980s by the Sea Lion Cancer Consortium, and we've looked at genetic factors, herpes virus, and the persistent organic pollutants. In this case, particularly the DDTs. There's been high usage of DDT in the US and the production, so those are the contaminants that we're worried about. And my colleague Karina Acevedo Whitehouse looked at the genetic factors. So she carried out a study to look at inbreeding using genetic markers to estimate this internal relatedness, which is a measure of inbreeding, and found that indeed the carcinoma animals were relatively more inbred than the trauma animals. But actually, when we looked at this in a little bit more detail, we found this phenomenon was actually driven by one particular microsatellite marker, one particular genetic marker called PV11. Now, these markers don't encode for proteins, they're neutral markers, but they are found within genes that do encode for proteins. And the PV11 mapped to one of these areas in the HPSE2 gene, which encodes for a protein called heparinase 2. And we know that heparinases are important in multiple carcinomas in humans and in animal models. And we found that this was linked with the carcinoma cases in the sea lions within a single homozygous PV11 genotype. So you have two copies, and if they're identical, if the alleles are identical, then you're twice as likely to have cancer than if you're a heterozygote, if your two copies are different. So we thought we might have evidence for a genetic basis for this carcinoma. But we're also concerned about the herpes viruses. And this is a widespread virus in this species. And again, we looked at sea lions with and without cancer and screened them for otorine herpes virus 1. And we found, with doing some detailed sequencing, that OTHV1 also has viral oncogenes within it, so cancer-causing genes similar to perhaps other herpes viruses that cause cancer, so Epstein-Barr virus and the Kaposi sarcoma-associated virus. And it's highly expressed in these sea lions with cancer, so the probability of having cancer increases with an increase in the viral load. So we have evidence for this as being a driver. We're going to finish up a study looking at all these factors, including the DDTs, to see how these multiple drivers are involved in this important carcinoma. Well, I'd like to finish with a story perhaps of scientific serendipity, and that's our study of leptin. And the nice thing about the Sea Mammal Research Unit is that you can get involved in all sorts of different studies with your colleagues. And one of the questions that we've been asking over the years was, how do animals know how much fat they have, what kind of condition they're in? These animals need to have a certain amount of blubber in order to be able to breed or to survive through a fasting period. And one of the hormones that's important in terrestrial animals is this peptide called leptin, and it's produced by the adipocytes in the fat cells themselves, and it signals to the brain to regulate body weight and food intake and energy expenditure. Well, when we tried the question was, is this the signal? Is this what role does this play in the seals? When we tried different antibodies that are commercially available and raised against other species, none of them seemed to cross-react. So it made us think that maybe the leptin molecule, leptin peptide, is different in the seals compared to other animals. And indeed, when we looked at the amino acid sequence here for the grey and the harbour seal, you can see the red letters there to show where that was different, and where the dots are, that means that the amino acids are the same. So there's no very, very little difference between the harbour and the grey seal, but quite a lot of differences between the dog and the human. And when we looked at that in a tree, certainly the seals do cluster with the carnivores as expected, but they're out on a limb here, and they're quite a bit different in their amino acid sequence. So that might explain why the reagents didn't work. But we thought, well, we'll just do a tissue distribution study and look to see where these peptides are expressed. So I ran this on a gel, looking at the different bands to identify the leptin. And we did indeed find it in the blubber, so lane C, in both the harbour and the grey seal, and also in the bone marrow, as expected. But then we found it in the lung. And this is where the serendipity comes in. This is very puzzling. We really never expected to find it there. And then we found a publication by John Torday, who had investigated the stretch-stimulated surfactant pathway in the lung. And he described its a process. So the lipofibroblasts, when they stretch, they're signaled through these signalling proteins to the alveolar type 2 cells, which then go on to produce surfactant. Now, surfactant in the lung is really important for lung compliance and to prevent the surfaces of the alveoli from adhering to each other. What John also found was that there's another pathway that's expressed just in the fetus, which involves this leptin molecule, and that signals to a leptin receptor on the AT2 cells. And this pathway is only seen in the fetus after birth. This pathway is switched off and is not used anymore. Because the fetus in utero has a collapsed lung, but when the baby is born for the first time and takes a breath, then it needs additional surfactant to unstick those alveoli surfaces. So you can see the surfactant there around the airspace. So without that additional surfactant, it's perhaps difficult for those airspaces to inflate. Well, what we know is many marine mammal species experience this lung collapse every time they dive through the pressure, and therefore leptin increasing this surfactant production. We know there's a high turnover rate of surfactants in marine compared to terrestrial species, so we hypothesise that this leptin pathway may be retained after birth. And when we looked at different sequences, again for the Weddell seal, another phosid species, and the California sea lion and porpoise and put that into a tree, by which time there had been more sequences published for this peptide. We found that there was actually evidence for positive selection on this arm going to the phocids. Well, what's interesting about this group is that they have the least modified of all the airways in the marine mammals. So many marine mammals over evolutionary time have adapted their morphology of their lungs. The cetaceans have large air sacs and don't really possess alveoli any longer. But the phocid seals still retain this link with the terrestrial cousins. And so it seems like this selection pressure exerted on this pathway is an adaptation which is unique to the phocid lineage. And you can see here in the boxes are the helices of the peptide, and the areas in red are where the differences are in the seal leptin, and you can see they're clustered in what's called the AB loop of the protein and in this E helix. And if we look at that in a diagram, so this is looking at the molecule from the front and the back, if you like, you can see here the AB loop and where those residues are different in the seal and in red here in the E helix. But indicated here by the black arrows are also the residues that we think are important in the binding of the leptin to its receptor. So to finish the story, we looked at the leptin receptor from a grey seal lung tissue and found that actually this was highly conserved. So it's almost identical in all the species that have been looked at. This is a diagram of that receptor, and this is the bit where the leptin molecule binds to that receptor, the CRH2 domain, and this amino acid sequence is highly conserved. There's a few differences here with the dog, the pig, and the seal, but those are synonymous substitutions, so it doesn't make any difference to the function of this receptor. So the hypothesis that it still retains the ability to bind to a receptor is critical in that structure. What we wondered was whether or not in future this might be an interesting therapeutic agent if it binds to human leptin receptor, but it has the ability, seal leptin has the ability to increase surfactant production. Maybe it might be useful in therapy for babies that can't produce surfactant themselves in respiratory distress syndrome. So we're hoping that maybe one day somebody will pick this up. But I think this is a good story of scientific serendipity. Don't ignore an anomalous result, because it might take you into an area that you didn't expect to go. What about future issues for these species in this area? Well, we've heard about the climate emergency. We have now been recently involved in a study where we found phocine distemper virus in the North Pacific. So before it was not found in the Pacific region at all. We think it's endemic in the ice seals, in the harp and hooded seals, and they're now moving across into the North Pacific in areas where the sea ice extent has reduced with the warming of the region, bringing this novel disease into the fur seals and the sea otters. And the worry is then that if the PDV then spreads down in the Pacific through the different species, maybe into the highly endangered Hawaiian monk seals, which may or may not be resistant to this disease. So that's an example of climate change having an impact on pathogen spread and occurrence. We have increased urbanisation of the coast. More and more people throughout the world are living in the coastal zone, and that's impacting from the land to sea. And we know this effect of increased nitrate and phosphate input into the marine environment is a driver for phytoplankton blooms, and particularly the harmful algal blooms. So I think we are likely to see a continuation of those toxins into the future. We have our legacy and emerging pollutants. We all heard about plastics and plasticizers, so that could be an issue going into the future. We have evolving diseases. We all know very well about coronavirus, and so there's land to sea transfer perhaps of these diseases, but also vice versa. Do these populations maintain zoonotic diseases, or are reservoirs for diseases that might be important for humans and for livestock, particularly, say, Brucella and influenza virus, which we know are circulating in some of these species. And we have these multiple stressors, so we know that there's other impacts that we are not able to take account of always, so the ocean noise and disturbance. So I think there's a multitude of studies and investigations that we need to do going into the future. Well, it's not been all about sitting in front of a computer analysing data. I think it was Frank that said you have to put embarrassing pictures of yourself in your talk. So I've had the satisfaction of working a lot in Scotland, but also on a subantarctic island, Macquarie Island, with elephant seals, with dolphins in Florida. I've been to the Antarctic working on Weddell seals, and of course my trip to the Galapagos Islands. So only remains for me to say a huge thank you to everybody. I have done none of this work, has been a solo effort. It's all been about teamwork and collaborations, and especially everybody at the Sea Mammal Research Unit. It's been a huge privilege and a lot of fun working with all the scientists and the support staff over the last 30 years. And so full credit goes to them. My PhD students, who've been great masters and undergraduate students, have all contributed to this work. My friends and my collaborators across the world have allowed me to get involved in lots of different studies and wheedle my way into their field work. And of course my family, especially my husband Jez. Thank you very much.
Principal, vice principals, colleagues, guests, ladies and gentlemen. It's now my very pleasant and enjoyable task to offer this short vote of thanks to Professor Hall for a truly enthralling account of her studies, which took us deep into the world of CSI and silent witnesses, from the unexplained deaths of mortuary technicians while she was in London, via the strange jaundice of the Epping area, to the unexplained sea deaths on the shores of Scotland, and eventually to the Californian seal urogenital cancer. It is clear that Professor Hall is a very passionate and insightful epidemiological detective with a deep love of her work, or as she put it, a strong interest in mass mortality. This passion, along with her expertise, has enabled Professor Hall to collaborate with researchers from all over the world, visiting such extreme environments as the Antarctic, the Galapagos, and of course Scotland, and we're profoundly grateful that she's chosen to settle here. Tonight, Dr. Hall has shown us how it is possible for a person to realise a rich and varied contribution to important epidemiological questions. Her skills, both as a communicator and a scientist, have brought to life for us here today the complexities and hidden depths that conceal what can only in hindsight be seen as simple truths of infection, behaviour and immunity, and how, when they're out of balance, can result in such stark and widespread disease. We are fortunate at St Andrews to have scholars such as Professor Hall, not only to keep a focus on clear and important questions concerning disease and mortality, but also to remind us that, as with her story of leptin, we should be open to serendipity and the unexpected result. The nature of epidemiology is that it looks at patterns. One unwelcome pattern that I know is close to Professor Hall's heart is that of climate change and the associated environmental disruptions that it brings. Professor Hall, I know that the insights you have provided here tonight for us will help remind us of the ever-important need for vigilance in advocating for our world and all the ecosystems that it houses. Today, we're honouring Professor Hall for the research she's undertaken and for all the lives her work has touched, even if they don't actually know it. Whether that be through policy reform, improved codes of practice, or through her work in predicting risks of infection and PCB exposure to cetacean populations. Professor Hall, I would like to thank you most sincerely for all of your contributions to the field, for helping to safeguard our seal populations, and for explaining this small subset of your work so clearly that even a simple person like myself can actually understand it. It now only remains for me to reiterate our thanks to you and to invite everybody to the younger hall to engage in some social behaviour and perhaps imbibe just a little moderate amount of neurotoxin in the form of some wine. Thank you very much.